So we'll discuss about bacteriophage lambda, gene regulation in bacteriophage lambda. These are bacterial viruses. Basically, they have very limited resources, and it will be really interesting to understand how genes are regulated in this bacteriophage. We'll take example of bacteriophage lambda. This is called as a temperate phase. Why it is called as temperate phase? Because it has two survival options. Depending upon the situation, it can infect the bacteria. After infecting the bacteria, it can kill it, produce progeny particle, and the second one is that it can join the bacteria, right? It has linear double-stranded DNA as genome. The size of the genome is only 48 kilobase pair. You can understand very limited number of genes. This is how bacteriophage lambda looks like. This is the double-stranded DNA, right? You have this head which is made up of proteins. This is tail and there are tail fibers, right? Now let us understand what are the two survival options it has. The two survival options it has is first one is called as lytic pathway. What is meant by lytic pathway? This bacteriophage lambda will infect the bacterium, all right? And after infecting the bacterium, it infects the bacteria and utilizes host resources to produce 100 odd progeny particles. So you understand what is meant by progeny particles. That means 100 copies of DNA will be produced and those DNA will be packaged properly into the coat. Means head will be formed, tail will be formed like that. And once they are perfectly formed, they are completely formed, then bacterial cell will be lysed. Okay, and those 100 progeny particles will be liberated into the atmosphere. Okay, this is a very, what you can say, a natural course that is adopted by all the organisms. You know, they utilize the favorable condition to perpetuate, to increase their population. So this lytic pathway, you know, goes in that particular direction. So during lytic pathway, all viral functions are fully expressed. Please understand, viral genes are expressed, okay, but bacteriophage lambda doesn't have its own resources. It doesn't have enzymatic machinery to replicate the DNA and to synthesize the protein. So what does it do? It utilizes the host machinery, the bacterium which it has infected, and the host resources also. And it will replicate the DNA, make some 100 copies, and then those DNA molecules will be packaged into the coat, and the progeny particles, once they are completely synthesized, then at an opportune time, this is very, what you can say, precisely programmed. The bacterial cell will be lysed, and these progeny particles will be freed into the medium. So this is one, what you can say, life option, survival option that is available to bacteriophage lambda. There is another one which is available to bacteriophage lambda, and that is called as lysogenic pathway. The second option is lysogenic pathway. This is option number 2, and since it has got two survival option, it is called as a temperate phase. In lysogenic pathway, what happens after infection? The phage will infect bacterium. Phage lambda infects bacterium. And after infection, you know, the phage DNA integrates with the host DNA. And this integration takes place by what is called as site-specific recombination. Site-specific recombination. And once it has been integrated, this phage DNA can replicate along with the host DNA. It can replicate along with the host DNA for infinite number of times, for generations. Okay, so please understand, this is the second pathway, that is the second survival option that is available to bacteriophage lambda. In this, what happens, the phage DNA, after the phage has infected the bacterium and the DNA has been inserted into the bacterial cell, it integrates with the host DNA by what is called as site-specific recombination. Okay, and after integration, it will replicate along with the host DNA for generation. It can remain there like this for generation. So phage DNA in this form is called as prophage. It is called as prophage. In the latent form, when it is integrated with the host DNA and it is replicating along with the host DNA, it is called as prophage. And the bacteria which harbors a phage DNA is called as lysogenic bacterium. Which harbors a prophage, which harbors a bacteriophage DNA, is called as lysogenic bacterium. What happens during lysogeny? Viral functions are suspended. Viral functions are suspended. They are temporarily turned off. They are suspended. They are temporarily turned off. Please understand that they are not lost. Viral functions are not lost. Rather, they are temporarily suspended. As long as the bacteriophage will remain in this form, in the prophage form, the viral functions will be temporarily suspended. They will be temporarily turned off, but they are there. Okay. And lysogeny, it confers one benefit to the bacterium, to the host, that bacteria becomes immune. Immune to further infection by the same bacteriophage. This is very important, that once it harbors a bacteriophage lambda, a prophage, then that particular bacterium becomes immune. The second bacteriophage cannot infect it. Okay, this is one advantage. As long as the bacteriophage DNA is there in the bacteria, that bacteria will not undergo further infection. It will not be susceptible to further infection of the bacteriophage of the same type. So harboring a bacteriophage DNA, harboring a prophage makes bacteria, confers bacteria that immunity, right? So this is an important point, and why it is so, we'll discuss that point a little later also. Now, these two pathways, lysogeny and lytic, they are beautifully interlocked. We really do not know right now as to what determines the option. What determines the option means whether to go for lytic pathway or whether to go for lysogenic pathway, What determines this particular option? Okay, so there could be two possibilities. One is the conditions. Conditions, both environmental conditions and nutrient supply. So obviously, when these conditions are favorable, environmental conditions as well as nutrient supply is good, then one would expect the bacteriophage, or for that matter any organism, to perpetuate itself, to increase its population. Okay, so probably that particular law may be applicable here also. We really do not know. This is one, sort of a guess you can say. And the second one is the genotype of the bacterium. So these two, what you can say, things, they will determine, they will decide whether the bacteriophage will opt for a lytic pathway, or it will opt for a lysogenic pathway. When conditions are tough, when environmental conditions are harsh, when nutrient supply is not very proper, is not very good, then in that condition, obviously, you can also think logically that there is no point producing more number of progeny particles. Okay, so it is better to remain there in the host and multiply along with it, I mean replicate along with it. Okay, it can remain in the host for uh, generations. Okay, what I was telling you is that these two pathways, they are beautifully interlocked. Pathways are interlocked, and we'll very soon see what is meant by that. Okay, and there is always a switch, lysogeny to lytic. Lytic. What is meant by that? That, as I have been telling in this lecture, that bacteriophage can enter into lysogeny, the DNA can replicate along with the host DNA for generations. Okay, but when the host become hostile, okay, when the condition, when the bacterial conditions situation, bacterial condition is not very conducive, okay, but the host itself has become hostile, then at that time, that bacteriophage, it can switch over from lysogeny to lytic pathway. Okay, so what is that condition? That also we'll discuss. There is nothing like lytic to lysogeny, but from lysogeny to lytic, this lysogeny can be terminated and the bacteriophage can enter into lytic pathway. The trigger for that is the host has no longer very, what you can say, conducive, very comfortable. It has turned hostile. It's no longer a safe haven. At that time, the bacteriophage will switch over from lysogeny to lytic pathway. All right. So this is the background that, which is necessary for understanding the further regulation in this. Let us understand the lytic pathway. Lytic pathway. So in lytic pathway, I used one sentence, is that viral functions are fully expressed. Viral functions, you can say genes, are fully expressed. But these genes are expressed sequentially. Genes are expressed sequentially, right? What is meant by this? The genes in Bacteriophage lambda, they are expressed in 3 stages. And what are those 3 stages? The first stage is what is called as immediate early. Genes will be expressed in stages. They are expressed sequentially. The first stage is immediate early. And there are two genes in this immediate early. The first one is called as CRO and the second one is called as N. These are the names of the genes. And this CRO gene, it codes for an anti-terminator, anti-repressor. It codes for a protein which is anti-repressor. And N gene, it codes for a protein which is an anti-terminator. Right? This is stage 1. In stage 2, that is called as delayed early. There are 4 sets of genes in delayed early. There are 2 genes, they are called as C2 and C3. Right? They code for positive regulator protein. C2 and C3, they code for positive regulator protein. There are 7 genes for recombination. For recombination. There are 2 genes for replication. And there is a gene called Q, which is again, which codes for an anti-terminator protein. Okay. So in delayed early, you have 4 sets of genes, C2 and C3, which code for positive regulator protein. Then there are 7 genes which code for proteins which are required for site-specific recombination. There are 2 genes, the proteins of which they are meant for replication, which facilitate replication of the bacteriophage DNA. And there is one gene called Q, which is similar to N. It codes for an anti-terminator protein. The third stage is called as late genes. Again, there are 4 sets of genes, or maybe 3 sets of genes are there. So you have 3 sets of genes here, not 4. You have 10 genes coding for head proteins. There are 11 genes which code for tail proteins. And there are 2 genes which code for bacterial lysis, which code for proteins which are required for lysis of the bacterial cell. So what you can understand, that depending upon the requirement, these genes, there are limited number of genes, they are sequentially expressed. In the first set of genes which are expressed immediately after infection is these 2, CRO and N. CRO is an anti-repressor and N is an anti-terminator. Once these 2 have been expressed and their mRNA has been translated, immediately there will be second set of genes which are called as delayed early, in which you have 4 sets, C2 and C3, which are positive regulator protein. There are 7 genes which are required for, which codes for proteins which are required for recombination. There are 2 genes which code for proteins which are involved in the replication of the phage DNA, and you have another gene called Q, which is required for, which codes for an anti-terminator protein. In the third, which is called as late stage, there are three sets of genes, 10 genes which code for different proteins which are required for formation of the head of the bacteriophage lambda. 11 genes, they will code for proteins which are involved in tail formation, and 2 genes which are required for, which codes for protein which are involved in bacterial lysis. So sequentially, depending upon the requirement, these genes are expressed, and this is very nicely programmed this thing, because lysis of bacteria will take place only after complete formation of the progeny particles. Okay. Now, this is beautifully interlocked with the lysogenic pathway. So in lysogeny, you have only one particular gene that is expressed in lysogeny, and that is called as CI. CI gene is only expressed in lysogeny, and this codes for repressor protein. Only one gene is expressed during lysogeny, and that gene is called as CI gene. I'll be telling you shortly. And it codes for a protein called repressor protein. How these two pathways are interlocked, lytic and lysogeny, this crow, it inhibits transcription. Inhibits transcription of the CI. This crow protein, what is its job? It is an anti-repressor. So what does it do? It tries and it tends to inhibit the expression of the CI gene. Okay, and whereas this positive regulator proteins, C2 and C3, as the name suggests, they are positive regulator protein. What do they do? They facilitate expression of the repressor protein, that is CA. So using these two, the two pathways are interlocked. One of the gene, CRO, its gene product, the anti-repressor protein, inhibits the expression of CI, whereas these two, C2 and C3 gene product, which are positive regulator proteins, they are involved in, they facilitate the expression of the CI gene, right? Now, let us understand. So I will now be drawing the genome of the bacteriophage lambda. Let us try and understand how the genes are arranged in the genome. This is the double-stranded DNA. Right in the center you have CI. Then on this side you have CRO, crow. And then you have a C2 here. On this side you have N. Right, you have C3. Okay, and this particular region is called as regulatory region. I'll label it a little later. And then you have recombination genes, 7 genes which are required for recombination. You have 7 genes here, and then you have 11 genes here, and then you have 10 genes here. Okay, on this side you have 2 genes O and P. Then you have gene called Q, and then you have 2 genes S and R. And what are they now? I will label it. These genes, this particular region is called as a regulatory region. This O and P, they code for replication. They code proteins which are involved in replication. This Q, I told you, codes for an anti-terminator protein. And S and R, they code for lysis, proteins which are involved in lysis of the bacteria. These 7 genes, they are for recombination. They code for proteins which are involved in site-specific recombination. These 11 genes, they code for tail proteins. And these 10 genes, they code for head proteins. These 7 genes, the names are also known. So if you want, you can write down, sorry. The 7 recombination genes are ATT at, then you have INT int, then you have XIS cis, then you have alpha, beta, gamma. I think there is one more delta here. So these are the 7 genes which code for, which are involved in coding for proteins which are involved in recombination. I am not sure about this delta. I'll check and let you know. All right. So this is the arrangement of genes in the genome of the bacteriophage lambda. Okay. So you can understand from this that it has really very few genes. Number of genes are very, very less, but it utilizes these whatever genes it has beautifully, and it has not just one, it has 2 life options, and it switches over, shuttles between these 2 life options, survival options. Let us see in little more details this regulatory region. So I am drawing this regulatory region in more details. Okay. So you have right in the center CI. You have CI in the center, and then there is, it is called as PM, means promoter for maintenance. So now you understand what is meant by promoter. Okay, so there is a promoter region next to the CI, and there is a subscript M which stands for promoter for maintenance. And then you have PROR. There is a promoter on the right hand side and operator within that promoter on the right hand side. And then you have CRO gene, right? And then you have termination sites here. There are 2 termination sites, T1 and T2. And then you have C2. And then you have, there is one more promoter called PRE, promoter on the right hand side for establishment. So you have to very carefully write this. Then on this side, you have PL, promoter on the left hand side, operator on the left hand side. You have N. Meeting will end abruptly anytime, and will continue beyond this in the next lecture. You have N, and then you have terminator sites here, and then you have C3 here. So this is this region expanded, right? CI, it codes for a repressor protein. CRO codes for anti-repressor. N codes for anti-terminator. And C3 and C2, they code for positive regulator proteins. Okay, so please understand, you have a promoter called PM, M for maintenance.